Second part of it will be fine. We'll now discuss with facilitated diffusion. What is diffusion? Please listen here carefully. Diffusion is a passive process. There is no expenditure of energy. Already you know what is diffusion. Movement of solvent or solid or gaseous substances from a region of higher concentration to a lower concentration till they attain equilibrium. Okay, so it's a very slow process. It's not a fast process. It takes some time, and it involves either solid, gases, or liquids. Now, when you are talking about solids, you can take the example of copper sulfate crystal. You know what is copper sulfate, commonly called as blue vitriol. Copper sulfate crystal suspended in copper sulfate solution, they increase in size because of diffusion. Okay, so that's an example of movement of solids from a region of higher concentration to lower concentration. Okay, that you can remember about in case of copper sulfate. That is also diffusion. You have to understand about. So gaseous movement within the plant body. Okay, they do not have any organs, like you can't find digestive system in plants. You can't find respiratory system in plants or excretory system in plants. But all those processes happen. Respiration is majorly by diffusion in plants, and also the entry of gases through the stoma. Okay, so remember about that. Now, the rate of diffusion, it might either be more or lesser based on these factors. The rate of diffusion is always towards the concentration gradient. What is concentration gradient? One level will be having higher concentration, another level will be having lower concentration. Diffusion is always towards the concentration gradient. Andre, it always moves from higher concentration to lower concentration and not vice versa. Against the concentration gradient, Andre, it is movement of solids, liquids or gases from a lower concentration to higher concentration. For that, some expenditure of energy is required. Okay, so that is against the concentration gradient, which we are not going to discuss about. Diffusion always takes place through the concentration gradient, from the region of higher concentration to lower concentration. Okay, and the rate of diffusion will be more if it is towards the concentration gradient. Second aspect, permeability of membrane. See, for these molecules to enter into the membranes, so you know the plasma membrane structure of it, fluid mosaic model, biphospholipid layer. They have proteins which are floating in this model, isn't it? Some of them are transmembranal protein. Some of them are superficial proteins which are on the top of the layer. You might have drawn that fluid mosaic model of plasma membrane. Visualize that. When I have drawn this diagram, this is the membrane structure which is made up of phosphobilipid layer. So they are selectively permeable. They do not allow all the materials to enter into. Cell membranes or plasma membranes are selectively permeable. So based on their permeability, more diffusion takes place. If they are impermeable, there is no diffusion at all. Okay? Then temperature and pressure also determines the movement of molecules through diffusion. Okay? So these are all the various factors which affects the rate of diffusion. One is concentration gradient. Always diffusion is more towards the concentration gradient and no diffusion against the concentration gradient, because diffusion is a physical process. Permeability of membranes. The cell membrane or plasma membrane is selectively permeable. It does not allow all the molecules to enter into them. Selectively permeable. Okay? So the impermeable particles cannot enter. So the rate of distribution of impermeable particles will be lesser. Temperature and pressure also is going to either favor the diffusion or decrease the rate of diffusion. Optimum temperature idre, there would be better diffusion, and lesser or higher temperatures there might not be diffusion. It might affect that. So please remember about it. Diffusion also depends on the size of the particle. If large particle is there, they cannot enter through the cell membrane. If they are smaller the particle, better their entry into the cell membrane. Okay? So it depends on the size of the substance. Solubility in lipids, they are made up of biphospholipids. hydrophilic substances cannot easily enter. What do you mean by hydrophilic substances? Water-loving substances. Okay. So here, lipid-loving substances can easily enter. That is what it implies. Solubility in lipids is rare, the diffusion would be better. Diffusion depends on two aspects. One is size of the substance and solubility in the lipids. Because cell membrane itself is made up of biphospholipid layer. You have to remember about that. Okay. Now, when should facilitated diffusion take place? Some of the substances cannot enter through the membrane because they are having hydrophilic moiety. That means they are more affinity towards hydrophilic. They are water-loving substances. For them to enter through cell membrane is difficult. And at that place, there is a facilitated diffusion takes place. See, I am a facilitator for you. In what way? You have knowledge in the textbook. I am transferring that and making it easier for you to understand. So I am a facilitator here. If you read it directly, it will be very tough for you. That's the reason you attend to the classes, isn't it? Now, when the hydrophilic substances, they cannot enter, water-loving substances cannot enter through the cell membrane. It is facilitated by certain proteins. Certain proteins helps them in their entry. Just like I am a facilitator for you, for information, there is a protein which facilitates this. So we call it as facilitated diffusion. But facilitated diffusion, please remember, it's not a active process because there is no expenditure of ATP. There is no expenditure of energy. Facilitated diffusion is a passive process and it is facilitated by proteins that are found on the membrane. So there is no expenditure of energy. But for which substances do they facilitate? Hydrophilic substances, which cannot normally enter through diffusion, they facilitate them. Okay? So membrane proteins have sites which facilitate this diffusion and they should have concentration gradient. Again, facilitated diffusion is not against concentration gradient. It is with concentration gradient. Facilitated diffusion also takes place from higher concentration to lower concentration. It is not against this concentration. Okay? And special proteins doesn't require ATP. So I have drawn a diagram here. This is the transport protein. Okay, this entire thing, pink colored one is a transport protein. This is the molecule which has to be transported. Now this transport protein takes it is outside the cell, it takes it towards the cell, inside the cell. So this is doing the facilitator role. The transport protein is doing the facilitator role. They are transporting the molecule from outside of the cell to inside of the cell. This diffusion is nothing but facilitated diffusion. So facilitated diffusion is specific and sensitive to inhibitors. It cannot take inhibitors inside. Okay. So what are porins? Porins are found on the plastids and mitochondria. They are proteins, transport proteins. What do you call this transport protein as? Porins. Transport proteins are called as porins. So now they have given an example. The water channels for conduction of water, they are made up of 8 different types of aquaporins. See, semi-permeable membrane, selectively permeable, they allow substances which can dissolve in lipids. But water cannot dissolve in lipids, alva. They require special facilitators, transport proteins, which we call it as porins. Aquaporins, 8 different types are there on the cell membrane, which help in the entry of water into the inside the cell from the membrane. So that is an example. Facilitated diffusion can be of 3 ways: symport, antiport, uniport. Symportally, 2 molecules are taken inside the cell in the same direction. Antiportally, 1 molecule enters, the other molecule exits from the cell. So it is in 2 opposite directions. 1 molecule enters through the porin, another molecule exits through the porin. So we call it as antiport type of facilitated diffusion. Uniport, only 1 molecule enters. So we call it as uniport. Symport has 2 molecules entering inside the cell through this porins. So accordingly, facilitated diffusion through the methods of molecules entering, there are of three types: symport, antiport, uniport type. In symport, there is two molecules, A and B molecules are entering inside. In antiport, A molecule is entering inside, B molecule is coming out. So they are in opposite direction. In uniport, only one molecule enters, A molecule enters into the inside of this cell. This is what you have to remember regarding facilitated diffusion.